So far in cellular respiration, we've covered the glycolysis pathway and acetyl-CoA synthesis, which are these first two lines, all right, which would get us a total of 12 ATP. Our final is going to be 32, so we still have a lot of work to do. And so we're going to start with acetyl-CoA synthesis, which should be review. Then we'll move through the citric acid cycle, and then we'll do the electron transport chain and actually synthesize all of this ATP. Alright, so let's look at where we've been. So we started with glucose, we went through glycolysis, and we ended up with 2-pyruvate. Now we're going to take those 2-pyruvate and convert them to 2-acetyl-CoA so they can enter into the citric acid cycle. Alright, so if we look, alright, so this is the matrix of my mitochondria. Alright, so this here is my mitochondria. Mitochondrion for singular, okay? And we've got our cytosol right out here, which is where glycolysis occurs. So what's going to happen is we're going to transport the pyruvate from the cytosol into the matrix. Alright, so we're going to cross the outer membrane of the mitochondrion and the inner membranes. We're going to cross those two membranes and come into the matrix. So when that occurs, alright, after it's transported, we're going to convert pyruvate to acetyl-CoA. So we're going to remove a carbon dioxide. So we want to remember pyruvate has 3 carbons. Acetyl-CoA now is a 2-carbon molecule. So the pyruvate then is decarboxylated, so we're going to remove the carboxylic acid group. Alright, we're going to do a redox reaction. We're going to transfer electrons from pyruvate to NAD+ to make NADH. And we are going to attach coenzyme A, and that gets us acetyl-CoA. Alright, so remember now we've made some NADH. Alright, so that is an electron carrier. It can go to the electron transport chain. Carbon dioxide, as we've been talking about, the electrons in carbon dioxide tend to have low potential energy. Alright, so this is just a blow up of the same idea. So what you want to be able to do is kind of describe the process that I just took you through. Now, we've made it through glycolysis to pyruvate. We've transitioned pyruvate to acetyl-CoA. So sometimes we refer to that, what we just did, as the transition step or acetyl-CoA synthesis. Alright, so you can think of it in either way. The net result is the same thing. Okay? The transition step makes sense to me in terms of leaving glycolysis and entering a different cycle, the citric acid cycle. So, but either way. Alright, so this is the citric acid cycle. You do not need to memorize the structures. Okay? So you do not need to memorize these chemical structures. Alright? And in fact, the only intermediates I require you to know the names of are oxaloacetate, citrate, acetyl-CoA. Alright, and then I expect you to know how many CO2 come off. Alright, so what happens in terms of carbon dioxide, how many NADH are made, and how many FADH are made, and how many ATP, and how they are made. Okay, so that's what I expect you to sort of gather from this is how many CO2, how many NADH, how many FADH2, how many ATP, and how they are made. All right. Okay, so I'm going to get rid of this and kind of clean things up because this is, the diagram itself is relatively complex and we will simplify it. Okay, so let's go through it and then we can simplify it. Alright, so the acetyl-CoA, which has 2 carbons, combines with oxaloacetate, which has 4. Okay, so we take a 4-carbon and a 2-carbon molecule, we combine them and we get the 6-carbon citrate. Alright, we then reorganize the citrate. Alright, so we go from citrate to cisaconate. From there we convert it to alpha-ketoglutarate. Now, so the first thing is we take 4 carbons, we add 2, that gets us to 6. Alright, now we're going to rearrange those carbons, right, so that we can oxidize them again. Here, this step, number 3, is the conversion of NAD plus to NADH, so you should see that as a redox reaction. Alright, and here we see a CO2. Okay, now that gives us our alpha-ketoglutarate right here. The alpha-ketoglutarate is then oxidized to succinyl-CoA, and as we look at this, NAD goes to NADH, so that's another redox reaction, and we have another CO2 that's removed. So we've already removed the 2 carbons, or 2 of the carbons that were part of our 6. Okay? Now, from succinyl-CoA, we're going to convert it to succinate, and as we do this, we're going to transfer a phosphate group to GDP, which is guanine diphosphate, very similar to ATP. Guanine diphosphate becomes guanine triphosphate, and then guanine triphosphate transfers the phosphate to ADP to make ATP. So this process is substrate level phosphorylation. Okay. Now, once we're at succinate, succinate undergoes a redox reaction. FAD is reduced to FADH2. Succinate is oxidized to fumarate. Fumarate is then rearranged into malate, and then we do one final rearrangement. All right, malate, which has 1, 2, 3, 4 carbons, right, undergoes a redox reaction. So malate is oxidized to oxaloacetate, and NAD plus goes to NADH. So we have another redox. So this is a redox reaction, and this is a redox reaction. Okay, so what you should have gathered from this then is that the NADH, the FADH2 are made by redox reactions. The ATP is made by substrate level phosphorylation, and 2 carbon dioxide came off of our molecules. All right, so this in terms of sort of being more simplified in how it would look. All right, so we're going to start with OAA, which is oxaloacetate, and it is 4 carbons. All right, that is going to be converted to citrate, which has 6 carbons. Okay, and then we have our acetyl-CoA, all right, that's going to come in and combine with our OAA, and this is a 2-carbon molecule. Okay, now at this point you take your line and draw it all the way around, okay, from citrate to oxaloacetate. And I want you to put some arrows on there so you can think of it as occurring in multiple steps. Okay? So, as we work from citrate around to the OAA, we want to remember that we are going to make 3 NADH. Right? We're going to have 2 CO2 come off. We're going to have 1 ATP. We're going to have 1 FADH2. Okay? And then we rearrange it back to OAA. Alright, so that's kind of the basic overall scheme of the citric acid cycle, which I often refer to as the TCA. Okay? Now, the CO2, this is about as oxidized as this carbon can get, right? We've removed as much electrical energy or electrons from it as we can, all right? Those electrons, the high energy electrons, are sitting over here with our NADH, or here. Okay? Now, if we look at the citric acid cycle, all right, we want to look at it also not only in terms of its ability to generate NADH, FADH2, but we also want to see it in terms of biosynthesis. So if we take alpha-ketoglutarate, for example, that intermediate, we can pull that alpha-ketoglutarate out of the TCA cycle, make it into glutamate, make other amino acids. We can even make the purines. Okay? If we look at oxaloacetate, we can convert that to aspartate. We can then convert the aspartate to pyrimidines or other amino acids. All right? Succinyl-CoA, we'll see that heme, as in hemoglobin, all right, and chlorophyll molecules use succinyl-CoA. Pyruvate can be used to make sugars and alanine. So these would be other sugars, right, not glucose. Acetyl-CoA, acetate, and then lipids. Okay, so what I want you to notice is that we've focused on this thing catabolically, right? We've been moving around like so. Well, that's great for generating NADH, ATP, but at other times we're going to need to pull these pieces out, okay, and these intermediates, all right, and we're going to need to synthesize other types of molecules. All right, so the question becomes, what do we do, right, with all of this NADH we've made? All right, so if I think about where it came from, so I made 2 from in glycolysis. I made 2 more, all right, because everything happened 2 times. All right, because remember I made 2 pyruvate at the end of glycolysis, so then we ended up with, this is per glucose, okay, 2 from the transition step, all right, and then we made 6 from TCA, all right, and we also have something called FADH2, all right, and we'll see how it behaves very similarly. All right, so we've got 10 of these things that we need to deal with, right, that we need to do something with. And we're calling these things electron carriers, right? So if NADH is equal to my electron carrier, all right, we can think of it as sort of like this. Right, it's got these electrons, okay? And what's going to happen is that NADH is going to deposit those, all right, onto another carrier, all right? So NADH is going to deposit those. So if I take, let's just say complex 1, which is going to be kind of the first stop in our electron transport chain, all right? We want to take these 2 electrons and we want to put them onto complex 1, okay? So the way this would look then is we would end up with NAD plus complex 1, but now complex 1 has our electrons. So this is a redox couple, in that what it means is that you have alternate states of these electron carriers. They are a couple. They have the reduced form, such as NADH, the oxidized form, NAD+. We have the oxidized form, complex 1, right? We have the reduced form, complex 1, that has the electrons. All right, so we want to be thinking of these sort of donators and receivers of electrons. Now at this point, complex 1 with the electrons, it now becomes the donor as we move along. So NADH was the donor in our example, okay? All right, so here's the electron transport chain, all right? What we want to notice is we want to think about this thing spatially first, okay? So we're in the mitochondria, all right? Remember, the TCA was occurring here in the matrix, okay? So we've got all of this NADH floating around, right, that we've made. The other aspect of our mitochondria is that there were two membranes. And so there's a membrane here, which would be our outer, all right? So our electron transport chain is in the inner membrane, and there's a space in between. So it's called the intermembrane space, or the space between the two membranes, all right? Okay, so how does the electron transport chain work? So NADH is my donor. It's going to donate electrons to complex 1. NADH is oxidized to NAD+, right, because it's given up those electrons. Complex 1 is now reduced. It takes those electrons and transfers them to coenzyme Q. Coenzyme Q, when you add the 2 electrons to it, it takes on 2 protons just to neutralize the charge, okay? So coenzyme QH2 is our carrier with the electrons. Notice the electrons have dropped in energy, all right, as they move through complex 1 to coenzyme Q. That energy, though, can't be destroyed, right? And so what happens is complex 1 is going to pump H+ from the matrix, where it's in low concentration, into the intermembrane space, where the H+ is in high concentration. So, as the electrons pass through complex 1, H+ is pumped from the matrix into the intermembrane space. The electrons are deposited on coenzyme Q. Coenzyme Q takes those 2 electrons and it migrates to complex 3. All right? Coenzyme Q is diffusible through the fatty acid tail. So it's a hydrophobic molecule, right, that can hang out amongst the fatty acid tails of our membrane and migrate as a shuttle. Now, ignore complex 2 for now. The electrons arrive at complex 3. Coenzyme QH2 donates the electrons and becomes oxidized back to coenzyme Q. Coenzyme QH2 donates the electrons to complex 3, and we have reoxidized it back to coenzyme Q, right? So I said that twice to make sure that you got it. Now, the electrons are at complex 3. The complex 3 electrons now are deposited on cytochrome C. So we oxidize complex 3, we reduce cytochrome C. Those electrons drop in energy again as they move from complex 3 to cytochrome C, and we pump protons from the matrix to the intermembrane space. Cytochrome C sits peripherally to the membrane, and it will migrate between complex 3 and complex 4, where it will donate the electrons to complex 4. Complex 4 then passes the electrons to oxygen to make water. All right? As that occurs, as those electrons are moved to oxygen via a redox reaction, H+ is pumped for a 3rd time into the intermembrane space. The other thing that occurs is that the oxygen, the reduction of the oxygen, right, the addition of electrons to the oxygen occurs in the matrix. And when those electrons get deposited on the oxygen, H+'s are consumed. That will steepen our H+ gradient. Okay? Now, that takes care of the NADH, right? That's how we're going to get the energy out of the NADH and sort of recycle it back to NAD+ so it can head back to the TCA cycle. FADH2, its electrons enter at complex 2. So the electrons from FADH2 are deposited at complex 2. Complex 2 deposits those electrons onto coenzyme Q to make coenzyme QH2. those electrons then follow the same path that we just went through. Complex 3 pumps protons as the electrons leave complex 3 to cytochrome C. Cytochrome C takes the electrons to complex 4. Complex 4 deposits those electrons onto oxygen. As that occurs, we pump protons again from the matrix to the intramembrane space. Now, what we've done is we've taken the potential energy from our electrons in NADH and FADH2, and we've moved that energy into potential energy in an H plus gradient. All right? We then allow those H pluses to come back into the matrix through an enzyme called ATP synthase. As the H pluses flow from the intramembrane space into the matrix, we synthesize ATP. So we use the potential energy of the H plus gradient to now make chemical energy as ATP. All right, so this is just another diagram, all right, that we just went over, and it's just trying to show you what happens as the electrons move through. All right, here is yet another one. The nice thing about this one is that we've got a nice high H plus gradient here. All right, and so now we're going to take that electrochemical gradient, and we'll remember that that is defined as electro referring to the fact that we have a charge gradient, lots of H plus, and chemical because we really are just talking about H plus, right? So it's hydrogen. So as they move across, we are going to synthesize ATP. Now, how does ATP synthase work? All right, so we call this process chemiosmosis. Okay, so chemiosmosis is when these H pluses, all right, so I'm going to make this one big, all right, migrate through, all right, the F0 unit of the ATP synthase. That causes this to spin, all right, so they take a ride all the way around and then they come off, all right. So we're going to make this thing as they push through here, all right, this thing's going to spin around. Now, that causes the F1 unit to spin as well. The F1 unit then, as it's spinning, it undergoes conformational changes that allow for the active site, right, so that would be our catalytic site, to undergo changes in shape that will cause the ATP and the PI to bind together and form the ATP. All right, and so the chemiosmosis part is this movement of the H plus. Okay, and so this ATP that is made by chemiosmosis, we refer to it as being made by a process of oxidative phosphorylation. Right, because the energy to cause the phosphorylation comes from the oxidation of glucose. Previously, we made ATP by substrate level phosphorylation. So this is a different process here. All right. So if we look at our summary slide then, we have glycolysis makes the 2-NADH, 2-ATP. Acetyl-CoA synthesis, 2-NADH. Remember, pyruvate, we made 2 of these, so everything happens twice. So we make 2-NADH, 1 each time through. Then we run the citric acid cycle twice to get a 6-NADH and 2-FADH2 and 2 more ATP. Then the electron transport chain and oxidative phosphorylation, which we just covered, all right, that gives us an additional 28 ATP. So the bulk of the ATP is synthesized by oxidative phosphorylation.